So while we start with this session, I request you all to please keep your microphones on mute. Now, talking about today's session, precision medicine is changing the way we understand, diagnose, and treat major life-threatening diseases. And this transformation is driven by high-throughput molecular data that is being collected from patients, animal models, and large-scale cell line experiments. And in this program, Bioinformatics for Precision Medicine, we would explore how the various omics data types generated in these studies can be analyzed and integrated to study the basic biology associated with the viruses and their hosts, cancer onset, and the development and outcomes as well. My name is Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community managed by Pine Biotech. So before we start with today's session, I would want to introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use accessible tools, and our mission is to make bioinformatics more accessible. Omics Logic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Omics Logic is an international program that is currently running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. This online program includes weekly sessions with an instructor, online tracking and review of student progress, access to all the resources and materials online for self-paced learning, access to expert guidance and problem solving, educational and research pipelines, and domain data analysis on our big data server. Let me introduce you to the program mentors. We have Dr. Harpreet with us. She has an expertise in the application of statistical machine learning techniques on biomedical data and the development of prediction tools and databases, and she looks after the curriculum development, bioinformatics, and data science course development. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals and collaborations in industry and academia. And over the past several years, he has been developing collaborations with universities as a mentor and project lead for Omics Logic Training and Research Fellowship. And Dr. Cipollino Chavez, he's a postdoctoral fellow at University of Chicago. He is skilled in bioinformatics and is an expert in glioblastoma. Now, I would want to invite Ilya Brodsky, CEO of Pine Biotech, to discuss further about the session. Over to you, Ilya.
Thank you very much, Sonalika. And great to have all of you here today. So today we'll talk about our first session for Bioinformatics for Precision Medicine. And I want to begin with a brief introduction to what precision medicine is. So maybe I can ask that question to all of you. Again, thank you for joining. I would like to ask all of you, I'm sure that many of you have the background and the expertise and the interest in precision medicine. How would you define precision medicine? So if you could use the chat to type in your answers, I would love to know what you think. So what is the definition of precision medicine?
Hello.
Yes, hello.
Yeah, a medicine which cure the patient with minimal side effects.
Okay, excellent, thank you. Praveen, Anushka mentioned that this is a method of disease treatment. Saran is mentioning that personalized medicine based on pharmacogenomics. Excellent. Ensign Asp is saying that this is the use of tools and approaches in a more personalized way. All right, excellent. So great answers, everyone. And I think that we can turn to a formal definition of what is personalized medicine and what is precision medicine, because there's actually a distinction between the two. Okay, excellent. Someone else is adding here. Chinmay is saying medicines engineered to specific genotypes as to enhance efficacy. Gerald, medical solutions specific to the genetic characteristics of an individual. All right, Vinay is saying customized drug to each individual. So excellent answers. Let me read out the formal definition. So precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability of genes, environment, and lifestyle for each person. This approach will allow doctors and researchers to predict more accurately which treatment and prevention strategies for a particular disease will work in which groups of people. It is in contrast to a one-size-fits-all approach in which disease treatment and prevention strategies are developed for the average person with less consideration for the differences between individuals. So as you can see, the personalized and the precision part of it are actually intertwined. And what's important is that after decades of research, we now know that the detailed genetic and other molecular information about a patient's condition can be and should be used to deploy effective patient-specific remedies. Unfortunately, the overall statement that we are describing here is very much a broad statement that lacks certain details. And those details is what we want to focus on in this program. But this is a good point for us to start this discussion, because ultimately we mentioned the genotyping. So we mentioned genes and specific genomic information. We mentioned lifestyle and other types of variable factors that can affect the treatment efficacy or outcomes, and can also help us detect disease early. So we'll talk about these aspects as well. And importantly, what combines all of these factors is that these factors come from data. So today, this data is superficial, based on symptoms that are categorized with clinical broad terms. And we are moving towards a more precise definition of these terms using molecular phenotypes and molecular data. So before we continue speaking about precision medicine, briefly about myself and about our company. We are a technology transfer company. The company originated at the University of Haifa in Israel, and the company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky, combined the vision for precision medicine and the significant role that bioinformatics plays in precision medicine, where the need to integrate multi-omics data is a very important part and a cutting edge of research that is of interest to many different clinicians as well as researchers around the world. But this research effort has been taken into the broader audience by our team. Our team includes people that work on curriculum design, that help design delivery methods to make this content available online. It also includes programmers and bioinformaticians that are skilled in analysis of various types of data. And overall, our mission is to make data science available for a broader audience. So the overall theme of the Omics Logic programs is that we are focused on biology as a data science, developing applications of high-throughput data analysis to allow participants of all backgrounds to utilize them and solve the need for data literacy in application to biomedical challenges. And so here you can see some areas, including oncology, infectious diseases, neuroscience, as well as agriculture. So to address this challenge, we actually have designed kind of the 3 pillars of what our programs are all about. So the goal of Omics Logic is to provide training in bioinformatics, guided by mentors and peer example. We want to grow a community that shares a passion for data-driven research and an appreciation for citizen science. And we want to help students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life of data science. And so we have been working on enabling research using these three things: big data analysis tools that are easy to use and train people to use them effectively, provide mentorship by building a network of mentors that are available for answering your questions and guiding your research, and then providing training. And so this program is one of the examples of a training program that is designed to provide you with very specific learning outcomes. And briefly, I will mention also how we will achieve that for those of you that are joining this program. So one of the outcomes that is very important in this precision medicine topic is the basic understanding of scientific literature that describes analysis of omics data. Through the lens of precision medicine, we will go through project examples and literature that will help you understand this topic, not from me, but from experts that have spent years researching these topics. And together we will learn how they have applied bioinformatics methods effectively to extract meaningful and translational ideas that are applicable in the clinical sense. We will also focus on basic processing steps for next generation sequencing data analysis with consistent accuracy, so independently you can conduct such analysis. We will perform exploratory analysis and generate a hypothesis from data, or data-driven research. We'll focus on that. And you will be able to think about the different categories and factors in your data to design contrasts for statistical tests and prepare data for machine learning. You will also be able to annotate found signal using biological databases and references, identify appropriate data sets for answering a biological question, plan your bioinformatics analysis from start to finish independently, and present a project proposal, including the rationale, the methods, scientific background, and anticipated results. So these are really the objectives for this program in the context of precision medicine, or how does bioinformatics play a role in precision medicine. And to help us with all of this, we have this network of mentors that will be accessible throughout the summer. We will briefly introduce each one of them throughout the next several programs that we have. And the summer training programs are designed to actually lead to a research project. And for those of you that are considering this path, we have multiple programs available that include precision medicine, SARS-CoV-2 data analysis. We have programs on data science focused on technical skills like coding and machine learning. And we also have a research fellowship program. And the research fellowship program is a special opportunity that provides you with hands-on support and a research-focused group of people that can plan and actually execute the analysis that they're planning and develop a research project in a short time. Now, for all of you that are already a part of the research fellowship or are planning to find a research fellowship that fits your needs, we welcome you to first join a training program like this one. And then in the future, as you might be interested to work more on a research project, you can join the research fellowship. For those that are working already on projects and would like to join a research symposium in August 21st to 22nd, we will have a specially designed Omics Research Symposium that is organized with our partners at the Louisiana Biomedical Research Network, Georgetown University, University of Haifa, and many others that will provide you with an opportunity to highlight your achievement through a poster session and presentation of your project, prizes, and also to learn about cutting-edge research and applications of bioinformatics, undergraduate, master's, and PhD programs in bioinformatics, including for international students. We will offer also a glimpse of what the recruiters and industry are looking for in those that are willing to apply for a position. How do you get into an internship, or how do you identify job opportunities if you have been trained in bioinformatics? And also, we will discuss some effective resources for training and education in bioinformatics and data science. And obviously, there will be prizes for the best high school, college, and citizen science research projects. So we're excited and we're looking forward towards this. And all of you that are joining this session, if you're a part of our training programs or not, you are welcome to join, and we will be sending out information on this soon. So now let me turn to bioinformatics for precision medicine. So this is our first session. And in today's session, we will talk about translational bioinformatics approaches that lead to effective outcomes and help address the gaps in precision medicine. And let's start by talking a little bit about the future of medicine. So what is the future of medicine, and where are we to actually facilitate some progress in this? So I think that it is obvious that to make these better decisions about patients and treatments and outcomes, there has to be aggregation of data, and there has to be an integration between lab data, research data, and data coming from the patients. And all of this data has to be presented to experts that understand the data in such a way that they can discuss the results of analysis in a meaningful way, and they can translate this information to practical decisions that they have to make, leading to an effective identification of areas of research that then could be addressed by pharmaceutical companies, biotechnology companies, and others that are involved in addressing the unmet needs. And so what we see this is as a combination of personal and precise, and there has to be a number of components that come together to make that possible. So beyond the general statements like we have discussed up until now, let's talk about how does this actually play out in different examples. So most people would agree that we can divide the diseases into two kinds: infectious diseases and non-infectious diseases. So infectious diseases are spread directly or indirectly from one person to another, caused by pathogenic microorganisms such as bacteria, viruses, parasites, or fungi. And non-infectious diseases are not caused by a pathogen and cannot be shared from one person to another. They're caused by environment, nutritional deficiencies, lifestyle choices, and genetic inheritance. And so to study these diseases, we can leverage data that is widely available. And what I wanted to do today is take a look at some of these examples to kind of identify what types of data and what types of analysis is appropriate for each type of data, so that we have a sense of what might be the source of information and the application of the information that we derive from such data. So omics data in infectious disease research can include viral genomics and evolution, and we all know that this is a very relevant topic today as we are in the middle of a pandemic, and actually not just this pandemic, but we live in an era of series of epidemics, starting from SARS-1, flu, as you remember, bird flu, Ebola virus outbreaks, rhinovirus is now again reappearing. So we live in an era of epidemics and pandemics, and understanding the pathogens that cause this in a precise way is a component of precision medicine or precision public health. We also understand now different drivers of infection and responses, and those include our microenvironment, including the microbiome, and immune responses and markers of the immune responses that we can detect in patients. And ultimately, when we speak about infectious diseases, in a lot of ways, we are talking about, again, extracting meaningful signal from the data so that it can be made available to decision makers that can look at the data in an intuitive way. So they have to be presented with that data in an intuitive way, but also they have to be trained to be able to ask questions, research questions, based on the data that they see, and lead to important research topics that can be addressed effectively in the lab, in the research world, and by translational industry, which means pharmaceutical and biotechnology industry, as well as the clinicians that can actually take some finding in that research realm and translate it to practice. So the other topic that we mentioned are non-infectious diseases. And non-infectious disease is a good example for us to start with, where molecular information and sequencing information plays a huge role is cancer. And as we all know, cancer is a growing concern. We're much more informed about cancer. And what's obvious today about cancer is that there's a huge difference between types of cancer, between the people that are affected with different types of cancer, and the drivers of cancer in the whole population around the world. And one of the clear signs is that we know now that the cancer is associated with aging. A lot of cancers are affecting older people. And so as they become more risk susceptible, we need to understand how do we prevent and how do we treat this cancer more effectively. And one of the things that is very important in this is the ability to identify cancer onset and stratify patients according to risk, according to outcomes, and make sure that we incorporate this information in the treatment plan. And one of those is to identify markers of cancer risk. So before we go on, I have a few questions based on what we just discussed today. So what do you think are some examples of omics data that can help us study these conditions, including infectious diseases and cancer? What is an example of omics data that is used in such research? So if you think about different types of omics data, maybe you're familiar with different types of omics data, you can write it out in the chat, or if you would like to unmute yourself and say it, that is also fine. So what is an example of omics data? Okay, so Saran is mentioning metabolomics. Dr. Sudha, transcriptomics. Anushka Jain is also saying transcriptomics. Okay, excellent. NGS. NGS can be used to generate multiple types of omics data. Okay, Dr. Sudha is saying genomics, metabolomics. Okay, GWAS is a method of analysis. That's good. Metabolomics. Okay, anyone else? What other? So we mentioned transcriptomics and metabolomics and genomics, 3. Any other types of omics data? Just based on what we discussed already. Epigenomics, okay, so that's 4. What else? Proteomics, 5. Lipidomics, 6. Metagenomics, okay, excellent. Excellent. I think we covered a lot of that. Epigenomics, excellent. Okay, what kind of information can we learn from transcriptomic data? Okay, so let's take one of these. What kind of information can we learn from transcriptomic data? So there's, okay, excellent. Gene expression. Okay, so what gene expression is the information that we extract from transcriptomic data? Excellent. What are some of the challenges with analysis of omics data? Okay, integration. So Fernando is saying integration. Zara is saying SNPs. So SNPs, I'm assuming you're referring to what kind of information. That is true. We can extract the dominant SNPs from transcriptomic data, but more likely you're speaking about genomic data. But what are some challenges with analysis of omics data? So Fernando already mentioned integration. That's an excellent challenge, right? How do we take these different types of sequences and we extract genomic data, transcriptomic data, and all kinds of other data and integrate them to understand what's really going on? Okay, what else do you think are some of the challenges? And Danilo is also mentioning isoforms, long non-coding RNAs, microRNAs. Okay, excellent, Danilo, thank you. So what are some of the challenges with analysis of omics data? All right, thank you. Fernando is saying computational requirements, right? This is big data. Now that we have thousands of patients and each patient is generating some genomic, transcriptomic, metagenomic data, now we run into a problem of computational resources. Okay, what additional challenges are there for, okay, data management? Exactly. I've analyzed this, I've analyzed that. I have some tables. Where did they come from? How did I generate them, right? All of the data management questions come to mind. Okay, great. Thank you everyone for your questions. Last challenge. Okay, this is not for today. Advances in next generation sequencing. So we talked about omics data, and a lot of the omics data is coming from a very specific type of a technology. So once it was discovered that the DNA encodes for genes that are then transcribed into RNA, that are then translated into protein, we now know that next generation sequencing represents an excellent opportunity to study these molecular mechanisms of regulation that lead to specific phenotypes. And ultimately, we even mentioned here proteomics, lipidomics, metabolomics. All of these are functional enzymes and molecules and proteins that define the phenotype. But whatever is encoded in the DNA and the way that it is translated into proteins and all of these elements, that is the machinery that we can study to understand what is really going on. And the unique part about next generation sequencing is that the cost of next generation sequencing has been dropping very steadily over time. So if you think back to 2001, we started from $100 million for the full genome to sequence the full genome. And 16 years later, in 2017, for the first time, the price of the genome, the whole genome sequenced, was just around $1,000. And so because of that, not only is this data informative, it can give us insights into what's going on, but it is also accessible and affordable. And it is approaching that threshold that makes it comparable to some of the other tests that are being done in the clinic already, and we've been using them for a long time. And so to really understand that, we can look at the history of genomic data, which started in the 1950s when the double helix structure was understood. And then very rapidly, if you think up until 2018, we've already had thousands of genomes that have been sequenced for hundreds of thousands of patients around the world. And so as you can see, the price, the reduction in price led to an explosion of omics data that is now available in research for companies in biotechnology and pharmaceutical industry, as well as in the clinic. And so to talk a little bit about the different types of omics data, we can mention that there are many different types of omics data that we can extract based on sequencing and to understand the relationship between genotype, or what is found in the sequence, and the phenotype. The first one is Phenomics. We know that cells function and look very differently. Tissues, they form different tissues. If you take a look at the morphology of a cell, it can tell you that there's a huge difference in the way that it functions between, let's say, liver and blood, right? I mean, think about it. One is a solid tissue and one is a liquid. One performs one function, another one performs another function. So obviously that DNA that contains information on how to make those cells includes all kinds of recipes that we can study and understand what is the potential of all of these genes to perform different types of functions. But ultimately, because of the phenotype differences, we know that not all of these genes are equally activated. And so we have to go beyond just what's encoded in the DNA, and we have to look at how is that DNA regulated. And so this is a growing area of research since about 2009, 2010. There has been a tremendous interest in epigenomic data because it reveals epigenetic regulation. How do these genes become more accessible? How do they change from one isoform to another isoform? And so that regulation is called epigenomics. That data like chronic immunoprecipitation, like whole genome bisulfite sequencing, and many others, microRNA, etc., can really reveal that mechanism of regulation. And then finally, we have transcriptomic data, which allows us to understand what has actually been transcribed from the messenger RNA and how can we study the various isoforms and transcripts that are being present in a sample. Now, ultimately, what does this lead us to? This leads us to a change in the way a clinician can look at a condition. So if you think of this iceberg as a condition, a patient that comes in and says, my head hurts, or I have a lump. And so on top, on the surface, what we know is that there's some clinical data that might be available. But beneath the surface, there are these layers of omics data that can reveal the molecular properties driving this top of the iceberg clinical phenotype that the patients are describing, or that we can capture with tests like blood tests and imaging and other types of traditional forms of generating clinical data. Now, what's underneath the tip of the iceberg? Underneath the tip of the iceberg, we have the transcription, the translation, and the result of that translation as the protein. And so what we know now is that we can take a look at specific proteins that are critical for us to identify patients that will respond to certain therapies, like the HER2 protein, that help us categorize specific patients into subgroups and understand a lot better why certain patients have different responses to treatment or have different prediction or different anticipation of recovery and remission. So one of the examples for these is the evolution of breast cancer classification. So we know that breast cancer is actually a complex disease, includes many different tissues in the breast that are affected, and they also have different properties. They grow at different rates. They have different success at treatment. And so when we think about breast cancer classification, we now can separate breast cancers into multiple subtypes of the actual disease, based on protein expression, based on gene expression, based on different types of risks found in the genome and different types of mutations that are found in the genome. And so one of the ways that we will use this example is to really look for how does the prognosis, response to treatment, stratification of patients, drug purposing, diagnosis, target discovery, and risk prediction are all components of precision medicine that we are all anticipating. And so here's a glimpse of how we can study cancer using these kinds of technologies. We can look at understanding of risk early on. So the earlier, the better we can address the problem that we are anticipating. We can also look at likelihood of response to treatment. We can look at different types of drug safety parameters better. And so we can optimize the treatment, prevention, and prognosis ultimately for patients, and hopefully make sure that this disease is bringing lower mortality and a longer, healthier lifespan for patients. But at the core of this problem is the integration of data. So at the core of this, we need to understand how all of these pieces of data, bioinformatics, chemoinformatics, clinical data, how do all of them tie together to provide us with a better sense of diagnostics and therapeutics. And the problem is that this is really creating a gap, and a gap in understanding as well as a gap in industries that are trying to address this gap. And so the way that I think that this data gap is disrupting industries, and why there's a growing need in people like yourselves that are learning bioinformatics and that are learning translational bioinformatics, is because all of these industries, which are huge industries, have to adapt to this new status, which is that everything is data driven and the data is becoming more complex. And so as we think about this, we really should turn to how does this information coming from research get translated to actual useful information, and how is its information being used? And so I think what we will do in this program is we will start thinking about how experiments that are performed in the lab with tissue culture and cell lines, how can they inform the understanding of cancer and a systems biology that we can study in patient-derived xenograft models and animal models. How do these insights can get translated to actual practical applications that are applicable to patients in clinical trials, where we can test drug responses and improve the outcomes, as well as to the clinic. And one of the primary ways that this is being done is actually not even in the practice realm, but first of all, it is in the realm of design. And the design has to do with data-driven instead of a biological hypothesis-based approach, right? So today, the information is top down. So someone that has studied this topic, a particular type of cancer, a particular type of a condition, is thinking and learning from experiments and accumulates this knowledge and then designs experiments based on their understanding. And that is hypothesis based. We take a textbook approach, we identify some important critical portion, and we design an experiment to validate. On the flip side of that is the big data approach. And the big data approach is really saying, I have all of these accessible data sets. I can sample them and sequence them and understand what is going on to start asking the questions. How do I address a particular variation that I have found? Or how does this affect patients that I know of? And so that is really what this program is going to be all about. How do we change our thinking to a data-driven approach to the research data sets that are available online, linking them to clinical information that we can also find online, and that link helping us to think of a big data approach to different types of conditions and diseases and patients that could ultimately benefit from better treatment, earlier prediction, and better prognosis of outcomes. Now, I want to explain exactly how can you benefit the most from joining the program by identifying specific steps that you need to take from this session up until the next session to get access to everything that you will need to practice the technical and the hands-on activities that we will do and to learn all of the material. And so to talk about that, I want to ask Sonalika to again walk us through how we can get access to all of the resources that are needed.
Thank you so much, Ilya. Let me share my screen. Yes. So first of all, I would want you all to sign up on the portal learn.omics logic. I would be sharing the link with you all. If you all have signed up, then just let me walk you through how to update your profile and things ahead. But if you have not, let's give you 2 minutes to please sign up. This is the link, learn.omicslogic.com. I would want you all to please sign up here. You can sign up either by creating your mail ID and a password for yourself, or also you can sign up via your social, but I would recommend you sign up creating a password for yourself. And while others are signing up, I would want to take few of the things. Those who have already signed up, please log into your profile. And I would want to know in the chat box if you have signed up by either sending a 1 or a yes, so that I know who all have completed their sign up.
So, Sonalika, if you could please go to the main page and just show from the beginning, once people get on the portal, how do they actually take the steps to make sure that they're registered? Thank you.
Yes, thank you. So you can sign up either via your ID and create a password for yourself or via social. And let me just show how you can do so via social. Thank you for letting me know that you have signed up. And those who have not, they can sign up with me because that is what you would be required to go further in the process for the program. Thank you, everyone. And now, as you all have signed up, like I see many yes, those who have signed up, I would want you to go through the to update your profile. And while I'm guiding the ones who have signed up to update their profile, others can sign up in the meanwhile. Yes. So those who have signed up, I would want them to update their profile and also link in their social accounts. Linking in the social accounts would help us feature you all with your progress on our social sites. And also, we have a lot many recruiters. So seeing your progress, we could also take your profile ahead and highlight them in front of the recruiters. That would be a great help to you as well. And please ensure that as a part of the program, you are a part of the Bioinformatics for Precision Medicine program. You must see a badge of Precision Medicine here on your profile. This is for those who have already registered and are a part of the program. And please ensure that you have a starter subscription accessed with your account. If yes, then please send a yes. Anushka, Gerald, I can see you have said a yes as well. Please check this and please let me know that please ensure you have a starter badge and also you have the badge for the program, and also update your profile picture and link in the social. Once your profile is updated and you can see your activity on the profile itself, and also you would be able to get the certificates once you complete the given number of assignments for the program. And also to go ahead with the projects, though we would also take you further to complete your project and upload it in the later part of the session, but this is where you can just look at your activity and get the certificates as well, and also look at the courses and the programs that you all have covered. So if you all have completed your sign up, Zahra says no batches, only starter subscription. Okay, Zahra, I'll look into it. And Zahra, are you enrolled for the precision medicine program? I'm sorry, we have not received your enrollment yet. So if you have, then please mail me at marketing@pine.bio so that I cross-check. I should not be missing out on any of the enrollments. I believe now everyone has completed their sign up, and if not, then I'll share the link again and you can do so meanwhile or maybe later. Now, this is the program page with which you have to keep a check on. I'll just share the program page link. This is the program page via which you have to register yourself. Now, if you hover over the program page, we have mentioned all the session details in the page itself, which has the calendar. So you would stay updated with all the dates of the session and also the session details on which day what session would be covered. And on the program page in the session details itself, you would have the Zoom link here, and also the recording of the session would be published on the program page after each session is over. So let's say after this session, the first session, I would be uploading the recording of the first session on the program page so that you can go through the recording if you missed out something and then take your doubts, right? And all those who have enrolled, they're already a part of the program page. So please make it sure that by tomorrow you can see the recording of the session. And those who have not yet completed their registration process and would want to proceed further with the registration, on the same program page, they would get the option of the levels beginner, intermediate, and advanced. Now, these levels we have segregated on the basis of the provisions provided. You can just go through all the provisions which would be provided as a part of the program for these 3 different levels. And those who are a part of an intermediate and advanced level, they would get to do a research project under a mentor, and they can we would be guiding them further for how their research project has to be covered ahead and how they have to like go on from finding a data set and then working on it, doing the analysis, and then finally publishing their work. So we'll take them through everything towards the mid or end, those who are a part of intermediate and the advanced levels. And as a part of the program, you would also get the access to a lot of asynchronous course material on the portal, and the courses to which the access would be given as a part of the program are mentioned here on the program page. And the certificate which would be given for the program is mentioned here, is shown here as well. And this is what the format of the certificate looks like. And as a part of the program, you would also get the access to the T-Bioinfo server on which different sorts of pipelines would be carried out and the analysis would be done. Our mentors would be taking you through the pipelines as well and how you have to go ahead with each and every analysis. Now, those who have enrolled, for them, it is to know that your login credentials for the Learn portal and the server would be the same. That is, you must be able to log in with your same ID and password as that of Learn on the T-Bioinfo server as well. And if you face any of the issues, then do let us know. We'll have a look. And this is the detailed syllabus, because I know you all must be excited to know about the syllabus topics would be for each and every session. So this is the detailed syllabus page. I'll share the link of the syllabus page as well. And you can go through all of the associated online resources which you'll get as a part of each and every session too. And to your knowledge, we are also there on LinkedIn and Facebook. I would want you to be a part of our LinkedIn and Facebook page so that you stay updated with all the upcoming other opportunities as well, and also stay updated with who is doing the best. Like I told you that we would be featuring your profiles with the performance on our pages, and we are there on LinkedIn by the name of Omics Logic. I'll share the link again, and on Facebook we are there by the name of Pine Biotech. And I'll just take a second more. We are at present live on YouTube, so if you would want to go back and look at some of the things which Ilya mentioned, you can go to the link and watch the recording. Over to you, Ilya. Thank you.
Thank you very much, Sonalika. So I am going to show, once you are logged in, so again, for those of you that have completed your registration, when you go to the program link, and Sonalika, if you could again post the program page, on the program page, you will find the information that is already relevant to what we are doing today. So right here, you will see the Zoom link. For example, for our next session, you will find the Zoom link here once we approach July 8th and after today's session is over. And after you are done with today's session, you can click on Complete Session. By marking this session as complete, you will start getting points towards your certificate. So you can now see that I have 1 out of 10 sessions complete. And as you go through these, you will also see for the completed ones that we have already done, instead of the Zoom, after the session is over, this will become a video. Now, as Sonalika mentioned here, you have the different resources, and down at the bottom, we also have a Slack channel. So if you have joined, you can go to Ask the Question, and this should open up Slack. If you already have an account, you should be able to open Slack directly, just like I'm doing right now. And if you don't have an account on Slack, it's a free account, and you get to join our community of all of the participants. So here you can see that all of you that have joined, you should be a part of this Slack channel. Now, on the Slack channel, we will post important information about practical assignments. If you have any questions, you can ask them there. We will be posting the recorded sessions, and all of the needed information in a timely way will be updated right here. So again, this is just a way for you to be connected to all of the different resources. And also you will find here a way to book a meeting with us or ask questions and also see your certificate. But all of this is around your personal profile. So really, as Sonalika mentioned, try to complete this profile as possible. Okay, so let me now go and talk a little bit about why this program is going to be focused on data and how we're going to address this problem. So the 3 things that our program is going to provide you with are the tools, the skills, and the ideas about how to apply these tools and data sets to different types of challenges. And ultimately, we're going to provide you with a number of examples of how you can take cellular omics data, understand the challenge in the integration, and then try to understand the relationship between clinical and phenotypic data with the different levels of omics data that we will describe in detail, including genomic data, transcriptomic data, metagenomic data, and others. And as you can imagine, the obvious challenge with this is that you are going to be faced with many different technical challenges that are going to be difficult for those of you that do not come from an engineering background. So here I would like to ask you a question about your background. Okay, so I know that we have participants with different degrees. So you could be a high school student or a bachelor student or a master's student or maybe a PhD or faculty. So if you could please post in the chat box right now, what is your level of education, your current level of education? High school, bachelor's degree, master's degree, PhD, or if you are a faculty. Okay, excellent, thank you. PhD, master's, PhD, faculty, master's in biotechnology, faculty, master's in computer science, excellent, master's in biochemistry, master's in biochemistry. Okay, I'll just give it a couple more minutes. PhD, master's in technology and bioinformatics. Okay, excellent. Okay, so one of the challenges in bioinformatics is that there are a lot of methods, algorithmic methods. There are a lot of different types of data, and there are also a lot of things that are not well defined. So they're data wrangling, data visualization, different types of approaches to scripting that require everyone to be aware of how to just deal with data, what's called data literacy. And I think on the flip side, on the engineering side, what is oftentimes not clear to people coming from an engineering background is how do these methods of computer software essentially address the problems in biology. And so this program is really designed to bring those two together, to take the biological terminology and the computer science / engineering aspects of bioinformatics and bring them closer together. And to do that, we want to make sure that everyone is familiar with some of the basic ideas about biological background, as well as the basics of computational and data science skills that you need to have. And to do that, we have designed a number of courses that you should start with. So let me share some of those with you. Okay, so if you go to the courses tab, you will find here some important coursework that you can start with as you are getting started with this program or any other program that we have. For those of you coming from an engineering background, we recommend a course called Bytes and Molecules. It will introduce you to some of the basic terminology and give you a sense of what is a mutation, what is gene expression, how do we use this information to answer biological questions. For those of you that are not familiar with bioinformatics, we recommend to get started with this course called Introduction to Bioinformatics. And that will give you some perspective on the applications of bioinformatics in general, like the introduction to big data bioinformatics. How do we start thinking with this data-driven instead of a hypothesis-driven research? How is bioinformatics used in healthcare and biomedical research? And then also, what is translational bioinformatics? So if you have just registered recently or you are now registering, please make sure that you start with these courses. Okay, so again, you create a profile, and once you create a profile, this is a free course, so anyone can take this. And once you have completed this course, again, you can click always on your name right here, and you can go to the course tab to see your progress with different courses. So by next time that we meet, please make sure that you have completed Introduction to Bioinformatics and Bytes and Molecules. So these are going to be important courses for you to complete so that we can all continue and move forward. Now, the other side of things is that you will need access to our big data analysis platform. Okay, so let me briefly explain how it works and what we will do using a short example. Okay, please go to this link, server.tbio.info. For those of you that registered today, you probably don't have your access yet. For those of you that have registered previously, starting from yesterday, you should be able to use your Omics Logic login details, your email and your password to be able to get in. So please let me know once you're logged in. You should be able to see your name right here, your email address right here under your profile. Okay, put number 1 if you can log in, or no. So number 1 or yes if you can log in, number 2 or no if you cannot log in. Okay, so Dr. Sudha, again, given that you have created an account on Omics Logic, not today. So if you just created your account today, you still don't have access. It takes up to 24 hours for your profile to be created on the big data analysis platform. But for those of you that have registered a while ago, yesterday or before that, you should already have your login. Okay, so Anjali, Praveen, Dr. Sudha, I am assuming you just registered today, correct? Anushka? Yes. So if you just registered today, you will not have access, but you can still follow along, and then tomorrow you can replicate the same steps. So your free account will come with this information. So let me briefly introduce you to what we can do on this portal. And to do that, I will take an example project that we will go through in our discussions. Okay, so Gerald, when did you sign up? Okay, so Gerald, if you can try, and Sonalika, maybe you could help Gerald. He should have his account set up already. If he doesn't, maybe Dr. Harpreet can help him. Okay, so once you're in, you will see that this is a big data analysis environment that you can use to try out a number of different approaches to analysis of big data. And instead of going into the main section, what we will do is we will open it up as a dropdown, and we will take at this cell line project, the first project right here. So once you're inside, you will see that this is a graph of different methods. Each box here represents an algorithm, and this algorithm has a logical purpose. That logical purpose is stated in the box above. So when we take raw sequencing data, we would like to clean this data. We would then like to map it on the genome, make sure that we are capturing the isoforms and recording them in the GTF file, mapping this information on transcript. So we know transcript abundance by quantifying expression levels, and then performing differential gene expression, or even just preparing our table of expression for downstream analysis. And so to illustrate how you can go from raw data to structured data, I will just demonstrate a brief step-by-step process where we start, we clean up the data with Trimmomatic and PCR clean. We then can map onto the genome, isoform construction using cufflinks, add that information to the GTF file using GTF processing, mapping on transcripts using Bowtie2T, preparing an RSEM gene expression table that looks like this. And then we can actually click on end to see the output files. And so here you will see a number of different outputs. So in a similar way, what we will do in this program is we will go through genomic data, transcriptomic data, metagenomic data, and introduce you to some of the common tools and methods that you need to understand and place them in the right position in the bioinformatics pipeline to get meaningful results. Does anyone have any questions about this before I go on? Okay, the second thing that you will be introduced to are some data repositories. So we will not just take data sets that we give you, but we will actually introduce you to repositories where you can find data independently. Does anyone here have any sense of where to find data about cancer? Omics data about cancer. I'll give you a hint. One of the repositories is right there on the screen. All right, excellent. CBio portal, TCGA, excellent. So what is TCGA? What kind of data do we have on TCGA? Leanna. Okay, Cancer Genome Atlas. So Dr. Sudha, okay, what kind of data do we actually have on TCGA? Okay, the answer is we have patient data. So there are a number of different tumors that exactly cancer genomic data, cancer transcriptomic data. So it's multi-omics data that has been integrated so we can easily search and find different types of data. But beyond that, there are also data sets on NCBI, for example, National Center for Biotechnology Information. So we will also go through some research papers that describe applications of cancer cell line and patient-derived xenograft models, so animal models, that we can use to find interesting insights like biomarkers that have a computational component to them and allow us to predict outcomes for different types of treatment response. So we will take a look at some of those data sets. In the TCGA data sets, we will take a look at the association between clinical information and genomic information, as well as other types of omics. And ultimately, we will also take a look at some infectious disease research challenges where we can identify important information about infectious diseases and the pathogens that cause them, as well as the immune responses. So as a result, you will find important information on how to perform standard analysis like phylogenetic analysis, gene expression, composition, analysis of alignment and variant calling, insights into structural biology, and how to integrate all of these data sets. So we will talk about those. And what I recommend to get started is to start looking at some of the important project examples that we will be using throughout this program. Okay, where do you find them? Again, you go to courses and you can start looking through some of the examples right here. For example, TCGA Liver Cancer Project on Precision Oncology, or COVID-19 origin and pathogenesis of SARS-CoV-2. So if you have an interest in cancer type data or in infectious diseases, you can find some project examples. Okay, so finally, I want to mention again that all of this information is going to go towards your own individual progress. So even though this is a program, it brings you together as a group. The objective is for you to grow personally, and we hope that you will be able to develop an independent project. So how do you go from learning about these skills and trying out different examples to actually developing an independent project? As Sonalika already mentioned, we have a system designed to help you go from learning to then application. And that will be focused on the aspect of developing a bioinformatics project. So we hope that this program will help you start, conceive of a project, and then start thinking about how do you actually conduct the analysis that you have planned. So if you're a PhD, you're already working in some kind of a project where you have omics data, you want to integrate it, this is not just going to be learning about bioinformatics, this is going to be, let's do bioinformatics, let's get this done, and let's apply these tools to a data set of your own. And for that, you need to have the advanced account and advanced registration for the program, or you can participate in the research fellowship program as a additional level to your subscription with this program, and then pursue your project independently. So before we leave, I want to leave you with this slide where you will find important courses that you can start with: Introduction to Bioinformatics and Bytes and Molecules, as well as some information about how to get started with bioinformatics in Python and in R, and how to design a bioinformatics research project. So if you're coming from a life science background, how do you actually think about selecting a topic, finding that data set, and then applying what you learn in this program to design an independent bioinformatics research program? And once you start making progress, you will see that on this page, you have some simple steps for you to get to a level where you can conduct a project independently with our support and then publish it on here as a featured project. And just to give you a sense of what that looks like, if you scroll all the way to the bottom of this page, you will find here a couple of examples: glioblastoma, COVID-19 drug repurposing, and lymphocytic leukemia will give you a good sense of what you can come out with as you develop these project ideas during this program. So that's all we wanted to share in terms of today's session. So Malika, I will pass it on back to you if you have anything to add before we close. And if anyone has any questions that I can answer, please put them in the chat, and we'll be happy to see you again next time.
Thank you so much, Ilya, and thank you so much, everyone, for listening so patiently. So today we discussed about the role of bioinformatics for precision medicine. And in the next sessions, we would be going through the practical aspects, and also we'll take you through the project samples and go forward with carrying out the analysis. And also we would be talking about next generation sequencing and big data analysis. And then we would go towards discussing the applications for infectious diseases, that would be viral and bacterial infections. And then also discuss about the Omics data, the role of Omics data in cancer research, be it genomics, transcriptomics, or epigenetics. And we would be waiting for you all to interact with in the next sessions. Thank you for joining today. So yes, so we have a few questions. Thank you, Ilya, for answering. Also, there was one question, I think it went unanswered. Let me take that. And you can ask the questions if you have any sort of in the chat box right now. We can answer them for you.
So just about the software, I want to clarify, if you are going to participate in all of the sessions, in future sessions, you will require some software like integrative genome browser. You will be able to browse VCF files and see how we can identify mutations that are present in some samples. So we will have some of that software that is described in the projects that we will discuss. And for those of you that are wanting to participate in a project with an intermediate enrollment, you can, but the intermediate enrollment does not give you enough time. So the training that will come first will prepare you to design a project proposal, but it will not give you enough time to actually complete your project. And so we recommend having the advanced subscription, or if you already have an intermediate enrollment, also consider joining the research fellowship program, which will give you 3 to 6 months to actually complete your project.
Yes, thank you so much, Ilya. And also, I was getting the questions from LBRN participants. So bioinformatics for precision medicine is not a program for the LBRN. It is not under the programs under LBRN. So if you would want to proceed your application for bioinformatics for precision medicine program, I'll just mail you all the details.
And yes, for those of you from LBRN that want to participate in this program, in addition to other programs that you're involved in, we can definitely take your information and share it with the LBRN leadership to see if they are going to provide some kind of scholarship for you. But we do need to have your details for that, and we need to know for sure that you are interested. And I know, Praveen, did you want to ask something? I'm sorry, I think you were interrupted.
Okay, sir. Okay. No.
Okay, no problem. nearby, if you completed a master's in biotechnology, you want to learn bioinfo without any paid program, yes, you don't have to pay. You can learn a lot about bioinformatics on your own for free. There are many resources online. We provide some of the free resources like Introduction to Bioinformatics. So please do take the course, Introduction to Bioinformatics, no cost, completely free. Just make a profile nearby and start learning. Okay, excellent. Thank you, everyone, and glad that all of you joined. Looking forward to seeing you again next time.
Thank you so much, everyone. I'm just dropping my mail ID. If you have any of the doubts, you can ask me. It's marketing@pine.bio, or you can also find me at mohit@pine.bio or marketing@omixlogic.com. Vinay, yes, I'll share I've added you to the Slack channel already. So please check your mail ID. You would get the mail from the invitation for the Slack channel. Thank you, Ujasvi. Thank you everyone for joining. We are looking forward to